Today we're going to be talking about a new disease that has entered our country that is an absolutely devastating disease. I'm going to share some background information and some experimental results that we've obtained at Kansas State, and there's as many questions as we have answers to some of this. So we'll start off. Porcine Epidemic Diarrhea. It's here, ready or not. And the reason I stated it like that is we probably had forewarning. We did have forewarning. We had a lot of forewarning and have had for years, but we've pretty much ignored it. So last year I was fortunate enough to be part of a scientific exchange, and I spent actually almost a year ago today in China. And we got to visit a number of federal, provincial, and university labs. And whenever I'm traveling abroad, the first thing I ask is, what do you have that we don't want to get? And every one of the labs that we visited, the first thing that they said was PEDv. They said it's a seasonal disease. When it shows up, it is absolutely devastating. And the old vaccines that used to work don't work anymore. Okay. We've kind of known that PED has been on the rise for quite a while, but we haven't really done anything about it. Why is that? So those are questions that we don't have the answers to, but we better keep asking those questions. How did it get here? Very important. We need to find the answers to these questions. All right, so the disease itself, you've probably all seen these types of pictures. I'll just go through them again. One of the things I love as a diagnostician, I get to work with people in the field. They share information, they share pictures. Dr. Matt Ackerman shared the following pictures here. Sick pigs, this is one of the first cases of PED in the U.S. I believe this is in Indiana. Obviously they're sick, they're covered in feces, sitting on the sow, trying to stay warm. On the heating mat, really, really severe diarrhea. All right, if you're basically a nursing pig and you get it, you're dead. If you're an older pig, it's not quite so bad. And the older you are, the less severe the disease is. Your survival is going to be much, much better. So feeder pigs, obviously they're soiled, covered in diarrhea. This is the type of diarrhea that you get. Mortality rates are considerably less, but they do get sick. They're going to shed virus and shed lots of virus in the feces, which is probably one of the main mechanisms of transmission. All right, so the virology folks here know that the coronaviruses are actually these guys right here and here, the peplimers. This happens to be a picture that I took when I was at the Diagnostic lab at South Dakota 30-some years ago. This is TGE, but TGE was going to look exactly like PEDv. This guy right here is rotavirus. And the point is, a lot of times it's not just one virus that these animals are infected with. And we'll get back to that after a bit. One thing that you always, if a country is free of PED and it breaks, the first thing they say, it is a pig-only disease. It does not affect other species and humans. There are no safety concerns. I think that's really important. Other countries across the Western Hemisphere are sharing that message now, because it's not just in the U.S. PED has been in Europe, originally recognized back in 1971. Since then, it's worked its way across the world. Korea, Japan, the Philippines, China, and as it's done so, it's managed to evolve. The word I had in China last year when I was there is the old vaccines don't work anymore. And the old vaccines that they were talking about were not the European versions, they were the Japanese and Korean versions. So this virus is evolving. Incubation, you know, just depending on the age of the animal, it shows up fairly quick. The literature really said that it didn't last all that long, but studies that were done at Iowa State, some of the data that I'm about to show you says that it'll last, we can pick it up for at least 28 days. I think Iowa State's got some data, 35, maybe 42 days, I'm not sure, but it hangs in there longer than we thought. Like I said, it's age-dependent disease. Nursing pigs, the mortality approaches 100%. If you're an older animal, it's not so severe, and actually it can go misdiagnosed. A lot of times the grow-finish animals, they're thinking, at least initially, they were thinking it was rotavirus infection. So laboratory diagnosis in older animals really is a useful thing because it can look like other diarrhea diseases. PED looks exactly like the old epidemic TGE that old-timers know and recognize. Not the endemic form, but the epidemic when it comes through on these younger animals. There is no cross-protection between these two different coronaviruses, even though the disease presentation is virtually identical. Okay, so if there's a single good thing about PED, it would be how the different laboratories and researchers and organizations pulled together to combat this disease right up front. So I'm going to be using and presenting materials that have come from other presentations from other groups, and I want to make sure that they get recognized right up front. So Dr. Roger Mean is kind enough to share some of his slides with me. This is a slideshow that he gave at the AASV meeting in San Diego a couple months ago. Actually, Iowa State was in the heart of this, and they've done really, really heroic work in identifying and getting this thing under control. So when Roger showed this slide at San Diego, I was really intrigued with it. The first cases that they really were paying attention to were these cases, and they were nursing pigs, and it was classical presentation. It looked like TGE. They're convinced that it was TGE. They ran their assays. It was not TGE. They double-checked and checked again and did some EMs, found out it was a corona. KJ and his graduate students worked all weekend long and ran the PCRs on a pan-corona assay, found out that yes, indeed, it was coronavirus. ISU and USDA both got the virus and sequenced it and confirmed, yes, indeed, we have PED in the United States. That was formally announced on the 17th of May. I think you guys are all aware of that timeline or close to it. The thing that I found exceedingly interesting when Roger presented this data is at Iowa State they had, I think it was 500 samples. Roger, are you here? Correct me when I misspeak, but they had 500 samples that were still in the queue, let's just say that. They hadn't been thrown away yet. And they went all the way back to December of 2012. And so they retrospectively went and started looking for the virus. And guess what? Their first case that they found, that they could document that was there, was the 16th of April in a grow finish submission out of Ohio. And so it clearly was preceding by a week to 2 weeks what they were seeing in the nursing pigs. So how long has it been here? Back in this point in time, we're only seeing a few cases of nursing pig diarrhea, the really high mortality stuff. But if you look at all the grow finish positives that were around back then, I mean, this thing had already spread considerably at that point in time. I was just fascinated with that data. All right, diagnostic specimens. Roger did a great job of putting these slides together. I couldn't do any better, so I'm just plagiarizing his slides but giving him full credit. Samples that you want to submit to the diagnostic lab: intestine, both fresh and fixed. If you want to attempt to do virus isolation, intestine and intestinal contents are your best source. It's much better than feces. Don't freeze this stuff. If you do, your ability to isolate goes way down. Feces, fecal swabs, oral fluids are all very, very good diagnostic samples for PCRs. Every lab has good PCRs, and they work very well. I always love antibody assays because I like to look at pre and post and to see what's actually going on. If you have evolving viruses that are escape mutants, you can run an antibody assay and figure some of that stuff out. So those are samples that they recommend. I would recommend exactly the same thing, or K-State does too. The assays, right now the most important assay that we have is the real-time PCR. Every single one of the diagnostic labs developed their own real-time PCR assays at almost exactly the same time frame. Within 2 weeks, basically, of the announcement that the virus was here, virtually every lab had their own real-time PCR up and running. It's a pretty phenomenal response. And I really take my hat off to the different molecular groups in those diagnostic labs for working so hard to get it. And a lot of them were tweaked assays beyond what was in the literature, so that's pretty good. Immunohistochemistry, that's a very, very valuable tool you'll see shortly. Antibody assays, the easiest one and most common one at the moment is indirect fluorescent antibody. EM is a valuable tool when your normal tests don't work. Virus isolation, it can be done. It's a numbers game. It's a lot of work. Some labs are more successful than others. I think Eric Nelson of South Dakota has probably got the most isolates of anyone that I'm aware of. Iowa State has done very good. The National Veterinary Service Labs has done very good. It can be done, but it's an awful lot of work. Sequencing is a tool that we all can do. When is it appropriate to do? That's the $64,000 question. And I just saw a talk from Minnesota that says that looks like maybe there's drifting of these viruses already, 2 separate clades. So that's interesting. Histopath. The reason I wanted to include this slide is as Darren Manson was doing a lot of this inoculation work, he was very kind in sharing. And when he got the immunohistochemistry of this baby pig that had been infected, he shared it with me. And I go, wow, I've never seen such a massive infection. And then he goes, well, you know why this disease is so bad? And I go, why? And he said, well, because baby pigs are born with mature enterocytes. That's just the way it works. And so these young animals, their entire GI tract is lined with mature enterocytes, and that's what the virus needs to replicate. That's why they have such an overwhelming infection. And you can tell, it's pretty overwhelming. This is what the small intestine will look like 36 hours later. All the villi are gone. These animals are dying of malabsorption. That's just really, really devastating and really quick. So they'll basically die of dehydration. All right. So key players in PED research. The National Pork Board is absolutely critical in the research efforts that have been supported thus far. Some of these slides that I'm about to show, they came from Lisa Becton, and she was kind enough to share them with me. The main groups that really jumped all over this and tried to get things under control would be the American Association of Swine Veterinarians and the National Pork Board. USDA was also very much involved. It was a good team effort. I would also say the diagnostic labs. All the diagnostic labs worked together with the samples that they had to share and help each other develop assays. So this was truly a team effort. But the Pork Board really put their money where their mouth was. They went to the main committee, whatever that is, and said, we need money to support PED research. What's the main committee called? Okay, well, so anyhow, they got the money. There was $450,000 initially. Then they doled that out to do some research. They went back a couple different times. I think they're up over $1 million right now for PED research. They recognize this as a major problem, and like I say, they're putting their money where their mouth is and trying to get this thing under control. Other state swine groups have done similar things, and also the AASV. So the industry has taken this disease very, very seriously, and they're doing everything they can to get it under control and understand what's going on. The Pork Board also puts out a lot of information and management strategies and things like that. So my hat's off to them. I was fortunate enough, my lab in Kansas was fortunate enough to get one of those grants. And our goal was to look at where the virus camps out in these infected pigs, how much is shed, is the virus maintained in these animals, and if so, for how long, what the antibody response is, and is this kind of like TGE? Is there an aerosol component to this infection? So those are the goals of what we wanted to look at K-State. We inoculated 4-week-old feeder pigs, and these are the preliminary results. Most of them are getting better. Oh, I didn't know there's a timer here. Better watch this. Okay, a lot of people, this is a really, really intense effort. I won't say how many hoops had to be jumped through to get this thing to go in the time frames that they wanted it done, but it was really, really intense. Folks in my lab, this is our molecular virology group, did a heroic job in getting samples turned around. Bob's lab, they furnished a lot of the people. We had to do this in the BRI, the Biosecurity Research Institute, under BSL-3 ag conditions. Bob's folks were already trained. I had a few of my folks that were trained. It was absolutely a heroic effort to get this thing done in the time frames that we did. And then most recently, Ying Feng has joined our staff at K-State. She's been working on some of the serology assays that I'll show you about that are pretty cool. All right, experimental, like I said, BRI, BSL-3 ag. Don't do it unless you absolutely have to. It's a lot of work. Good containment, but yeah, it's a major undertaking, let's just say that. Experiment, but because it was done at the BRI, the number of animals that we used was relatively small. 33 animals, naive 3-week-old pigs when they were brought into the facility, divided into 3 groups, inoculated, and they're inoculated the oral nasal route. We had 5 contact controls that were commingled with them, and then in the same room, in the same airspace, we had another pen that were the aerosol controls. The challenge that we used was true feedback material. This is actual material that was used to feed back sows following an initial break to take advantage of a controlled infection to induce immunity to try to get the disease under control. It's ground up guts is what it is, if you're not familiar with what feedback material really is. It had a CT titer, a real-time PCR titer of 22, which is quite a bit of virus. When we tried to do virus isolation of it, we could show that the virus could infect cells, but we could not serially pass it. The pigs were 4 weeks of age at the time of challenge. They got 5 mils in the nair and 5 mils orally. That's how we challenged them with that material. Experimental design, the study lasted 43 days. We wanted to get some samples for antibody response. Pretty loaded up front for sample collection and shedding information. We collected blood, nasal swabs, fecal swabs. We sacrificed some animals. Initially we knew we'd find virus, but we wanted to see how long we could find it, and we collected quite a variety of tissues. I'll go through those after a bit. And then we also collected oral fluid samples. Just a couple words about the BRI and the room setup. So this plastic sheet is a partition that separates the infected animals and the clean animals. So its purpose is to just prevent mass splatter, water spray, that type of thing. It's open at the top, and if you look This area right here is the front part of that partition, and there's plenty of room for air to circulate around. Obviously, in a BSL-3, the airflow cycles are pretty high anyhow, so there's a lot of air movement there. Divided into the dirty side and the clean side, and this is the normal garb, and you're showering out as you go through this whole procedure. It's quite a bit of work. Oral fluid collection. The more pigs you have in a pen, the better the oral fluid collection works. 5 pigs in a pen doesn't work quite as well, at least initially. They're not competing for it as much. Lots of open space. This is not normal production densities. Diarrhea. The pigs, the first clinical sign that we saw was vomiting in a small number of pigs. Was like 2 days post-inoculation? 3? Okay. Obviously, I wasn't there. So 3 days post-inoculation, we had some vomiting, and then the next day they broke with diarrhea. The diarrhea that we observed was clear cut, but it wasn't really that significant. Cow pie, maybe a little bit looser than cow pie, but I mean these pigs, they were infected, but not dramatically so. Immunohistochemistry on the small intestines from some of these animals. You can see where the infection occurs, those tips mainly. And so these samples were actually done, the IHCs that I'm showing here were done at Iowa State. We didn't have our assay up and running. They had a very, very good assay. Sent it to them. They did it for us. Sharing of labs, really important. Okay, like I say, histologically, what we saw, the lesions were mild. Dr. Neufeld said minimal is too easy. They were more than minimal. They were mild lesions. So if you're a pathologist, that's a discriminator. But they weren't all that bad. One of the things, a major part of our study is where else is this virus located in the pig. So we collected, at those time frames I indicated earlier, we collected turbinate samples, tracheal samples, lung samples, bronchial lymph nodes, a variety of internal organs as well, and we tested them for the presence of virus. Once again, we had to use immunohistochemistry because our in situ hybridization is not up and running. But we sent those off to Darren at Iowa State, and he read all the IHCs, and the only place that they found virus was in the intestines, small intestinal samples. No virus anywhere else. And then I said, huh. And he goes, well, Dick, you know, that's exactly what we saw on our own pigs that we inoculated. So that's a strong suggestion that this is only replicating in the GI tract, which is different than what you'll see with TGE. Shedding, okay. So fecal shedding, that's the inoculated animals, the contact controls, and the aerosol controls. All right. A day after they were inoculated, we couldn't pick up any virus at all. At day 2, we found it. Contact controls and inoculated animals, their shedding patterns are pretty darn close after day 2. And fecal shedding is pretty high. Aerosol detection, or not, excuse me, nasal swab detection. I originally used the term shedding, but based on the data as we gathered more and more of it, I'm thinking that's a misnomer. I'm thinking it's probably nasal contamination that we're picking up with nasal swabs because there's no evidence of replication in the respiratory tract. We can pick it up. It parallels exactly what the fecal shedding is. And like I say, the CTs are pretty high, so or pretty low, lots of virus being shed. The aerosol control, there's no evidence of fecal shedding in the aerosol control group, but in the nasal swabs, we're having some positive signal. What's going on there? But once again, no evidence of fecal shedding, no clinical disease, no anything. It raises some flags. So if we look at individual animals, I haven't found the best way to do that yet, but this is the average CT of a given animal over a specific time frame. And that will show you, so the shorter the bar, the more virus is present in that animal. That's about all you can take out of this. But it's a good way to show individual variation. And the inoculated animals, there's considerable variation there, less than what you see in the aerosol controls. No evidence at all of fecal shedding in any of the aerosol control animals. That is not the case. That is not the case with nasal swab testing. We have two animals that are clearly showing the presence of viral nucleic acid in their nares. Yeah, the inoculated animals, you're finding it, you would expect that. This is unexpected. Or was it? We'll get to that in a minute. Viremia, this is also another one that's kind of interesting. We look at blood samples and look for the presence of virus in the blood. This is the group average, CT. And clearly, the inoculated animals and contact controls had detectable viremia over this relatively brief time frame. Then when we look at individual animals, what we saw is the vast majority of them did not have a detectable viremia. Only a few animals did. And those would be the ones that are the shortest bars. Now, the thing that's kind of interesting there is there's a tendency for those animals to have the longer shedding periods. No evidence at all of viremia in the aerosol control group. Oh, I didn't even notice that. Okay. Oral swabs, same pattern as what we saw with the fecal shedding and the nasal swab shedding. So it's a very, very good sample, very easy sample to collect and get some profiling data on. But very nice. Aerosol animals, there's only 5 animals in that group, and they were kind of dumb pigs, it seems like, because they didn't want to chew on the rope till about 4 days into the study. But once they did, guess what? We're showing positive signal. They're PCR positive. So we thought, this is really interesting. No clinical sign, positive PCR, oral fluids, nasal swabs are positive on some of them. What's going on? So then we decided, well, we better look at the room, how much nucleic acid, how much virus is being shed into the room. And when we did that and actually swabbed the room on the inoculated side and on the control side, both sides of that plastic barrier, we found virus or viral nucleic acid. It was in the vitros that we used to bleed the pigs. It was on the walls. It was inside the pens. It was inside the feed bins and both sets of animals. So there's a lot of viral contamination. Whether that's infectious virus or not, we don't know, but it's certainly PCR positive. And at about the same levels as what the oral fluid samples were in our control pigs. But once again, no infection. IFA, so now we just switch gears to antibody assays. We have an IFA assay up and running at K-State. It's a nice assay. We like it. The order of magnitude as far as positivity seems to be pretty high. One obvious difference between our assay and Iowa State is ours, the dynamic range seems to be higher. I need to work with KJ and figure out why that is. I would expect very similar results between the two labs, but it is what it is. So we provided our samples to Ying, and she's working on 2 other assays, serological type assays. This is a conventional ELISA with MP as the target, And then, and as you can see, the profile there is virtually identical to, It's generally the same as, how's that? As what we see with the IFA response. And then she's also working on a Luminex type assay that will eventually be multiplexed with a variety of other swine pathogens, so you can actually herd monitor responses. So these assays are pretty much in agreement with what we see with the IFA assay. We hope to have SN results available by the end of next week. We're getting that going. The virus, the PEDs, we can initially isolate almost everything that we attempt to isolate. It's just serial passage of these viruses. We have not been successful. Like I say, others have. Eric has got the most number of field isolates that I'm aware of. So it's just a matter of keep trying and trying. So the virus that we have actually came from Dr. Sabrina Swenson at the National Veterinary Service Labs. Sabrina is very generous in sharing all these reagents, the isolation methods, the viruses with everyone. Once again, that's a real plus for the PED research effort. So these are syncytia that you would see. This is a crystal violet stain showing the perinuclear arrangement around the syncytia. IFA. So, I mean, once you get it going, you can work with it. Doesn't grow to really, really high titers, at least the stuff that we have, but it's usable. Gaps. We really, really, really need to understand transmission. There's been some really good work out of Minnesota showing truck transmission and fecal contamination. I believe it. There's a lot of virus in the feces. But there's also some evidence of aerosol transmission out in the field. And our laboratory data, both mine and some of the stuff that Darren did, saying, well, maybe not. We need to define the age differences in PED. Duration of shedding, persistence, is that a function of age at the time that the animal is infected? What are actual clearances? The disease patterns that we see in younger animals versus older animals, the leakage of virus into serum, things like that. We need to understand that better. Obviously, we need to develop better cell culture systems so we can get this virus under control, develop vaccines for it, classical vaccines. The serological assays, they're actually coming along quite nicely, so we'll be able to have better prevalence assay. And also intervention testing. So how much antibody did I get as a result of feedback? Or I fed back in June, how long will these antibodies be good for? So there's a number of serological usages that will be beneficial to the industry there. And last but not least, we need a good vaccine very, very badly. Aerosol transmission, trucks. Trucks are probably the best documented way of spreading this virus around. Aerosol, we had early on, we heard a lot of rumors out of Colorado that there's probable aerosol transmission, seemed to follow the wind patterns. Some groups said, no, I don't believe that. And then 2 months later, they broke and they go, oh my God, it's real. So there's probably something to it, even though it might not be a true aerosol infection that may be spread via the wind. If you consider, say there's pretty well documented, there's at least 10 to the 8th virions per gram of feces. Well, you get a break in a big production unit, how many grams of feces are there? And, if the wind starts whipping things up and blows it around, and it blows to a sow farm that has baby pigs, and you can just get a little bit, all it takes is 1 or 2 pigs to get infected and then amplify it. It might be an ingestion type amplification, but a wind spread. That's my suspicion. It has a unique ability to spread, and it's a seasonal disease. That's what the Chinese told us, and boy, it sure is. We're breaking all over the place right now. Kansas had its first real outbreak just on Monday of this week. And we expect the whole northeast end of Kansas, northeast, north central end of Kansas into Nebraska to just go down. Lots and lots of pigs are going to be dying. Hot spots, well, the darker the color, the hotter the spot. And this is current data as of the 29th of November. So the incidence, so these bars, initially when it broke in May, April, May, up, leveled out over the summer. And this is the data as of last week. Clearly on the rise. And it's the season for it. The AASV has good recommendations. Look them up. Vaccine production, basically grind up pig intestines. Wearing blenders work, but garbage disposals work as well. Problem with that is what else you bring with them, what other things you can infect. Lab data versus field data, they don't necessarily agree with each other. Water transmission, that's also one that's come to discussion. PCR positive water samples. We've, at K-State, have shared a number of our samples thus far. We've shared our complete serum sets with 5 different laboratories for assay development. That's a little over 1,200 samples that we've distributed across the nation. Oral fluids, same type of thing, not quite as many.